In our last video, we talked about what is DNA and what are nucleotides, and so continuing our understanding of the structure of DNA, we come across this American biochemist whose last name was Chargaff. He ultimately found a pattern in nucleotides. He found that the amount of A and the amount of T and the amount of G and the amount of C in a given species genome was different from all different species. So we might have 25% 25% G's, and a dog might have 20% G's or something like that. And so that could possibly be a way to tell species apart, was by how many of each nucleotide they have. And he also found a pattern which now becomes Chargaff's rule, which was that the amount of G equals the amount of C, and the amount of A equals the amount of T. And ultimately, this was proved by Watson and Crick, who are, of course, famous for discovering the structure of DNA and making the first 3D model of a DNA double helix. They were influenced by someone named Rosalind Franklin, who had taken some X-ray crystallography pictures of DNA sort of showing its double helix form. And while Watson and Crick were awarded the Nobel Prize for their discovery, Rosalind Franklin was not. And usually when I ask students, why do you think this is, they will all say, because she was a woman. And that's actually not the case. It was because by the time they were awarded the prize, Rosalind Franklin was deceased, and the Nobel Prize is not to be awarded posthumously. So anyway, what Watson and Crick sort of discovered was that there were two strands of DNA in our DNA double helix, and that hydrogen bonds occur between the bases to bond them together. So between A and T that actually match up, there are hydrogen bonds. There are bonds between C and G that match up. And so base pairing rules was that A always pairs with T and G always pairs with C, which of course supports Chargaff's rule in that the amount of G will always equal the amount of C, and that makes sense now because they will always be found together. And so that is our double helix structure of DNA.